Good morning. Welcome to our webinar on the LS and VS automated assays. We will first start with introducing BaseScope technology, then introduce BaseScope assays for the automated platforms on the Discovery Ultra and Leica Biosystems Bond RX. We'll review the product configuration and data for these two automated assays. Then Courtney Anderson will discuss various BaseScope applications. ACD is pleased to introduce the BaseScope assay, which is an expansion of our RNAscope technology. This is a novel reagent kit and probe combination for detection of exon junctions and short sequences. The BaseScope platform uses probes that are designed between 1 and 4 ZZ in length for single molecule detection. It is both a singleplex red assay for both manual and automation, and also exists as a duplex red and green assay for manual only. BaseScope is a proprietary in situ hybridization technology based on a unique probe design and signal amplification. This platform has a sensitivity to detect most genes in situ as well as simultaneously. Just as any in situ assay, the BaseScope assay involves permeabilizing properly prepared samples to allow complementary oligo probes to bind to the RNA target of interest. Specific signal is amplified using our next generation signal amplification tree and our double Z probe system, which is subsequently detected and quantified. The assay is extremely universal as well as rapid and scalable. To design a specific probe of your target of interest, ACD requests that you provide us either the accession number information or the exact sequence of the target. You could even indicate your region of interest for targeting with start or stop positions. Our bioinformatics scientists channel this information through the proprietary ACD alignment algorithm to select optimal sequences to ensure target specificity and uniformity of oligo hybridization in the assay. Our algorithm evaluates the sequence composition of each oligo in the pool to ensure no cross-hybridization with non-target RNA under stringent BaseScope conditions. So how does the technology work? BaseScope probe design is the key. We use a double Z probe design, which is very similar to RNAscope. There are 3 components of the Z. The upper region is the 14 base pair sequence, and that is the region where the pre-amplifier and the cascade of the amplifications will occur. A spacer region is in between the base and the other end, allows flexibility for probe binding efficiency to the mRNA. The lower end region of the Z binds directly to mRNA. This lower region contains 8 to 25 base sequences that is specific to your target. These are usually in pairs. In pairs, the upper region of the Zs will provide 28 bases for the pre-amplifier binding. Both Zs bind adjacent to each other to your target region from the lower region. The yellow line represents your RNA. If your RNA is intact under ideal permeabilization conditions, the assay will hybridize up to 20 Z probe pairs to your RNA. But for many samples, such as cancer FFPE samples, your RNA could be degraded or fragmented. The simplicity of this design, relatively short probes bind to the target, allows for binding of even short pieces of RNA. What our scientists have demonstrated in a lot of experiments is that we just need 3 ZZ pairs of these to bind to the target in order to detect a signal. Even for fragmented or degraded RNA, which are most likely to have migrated, they're usually still localized to each other. You should see a single amplification signal for each single molecule. The growth and adoption of RNAscope is best exemplified by the number of peer-reviewed journals. We had our first publication in 2011, and since then there are over 1,300 peer-reviewed publications using RNAscope in higher-tier journals. This slide summarizes all of the assays that are currently available from ACD. We have the chromogenic singleplex brown or red assay and duplex assay on both manual and automated platforms. The multiplex fluorescent assays allow to detect up to 4 targets simultaneously in a single tissue section. If your target of interest is shorter than 300 base pairs, it is below the RNAscope detection limit, and BaseScope would be the right choice for you. Currently, BaseScope is available on all platforms. The new addition to the family is the BaseScope Duplex, which is currently in early access. And now to introduce our LS and VS automated assays, which is a single-plex red assay. BaseScope VS universal assays BaseScope VS and LS assays can be run fully automated on both the Ventana systems for the Discovery Ultra and the BondRx from Leica Biosystems. BaseScope assays are optimized for FFPE tissues and leverage the red amplification kits from Roche Tissue Diagnostics or the red refined kit from Leica Biosystems, and can be visualized with any bright field microscope. Some of the benefits that are enabled with the LS and VS BaseScope assays. Both assays being fully automated allows for one to screen multiple samples overnight. LS and VS BaseScope assays are easy to use, not just due to the convenience of automated staining, but the assays have been customized so that the pre-treatment conditions have been optimized with minimal user intervention. And lastly, leveraging the high throughput of the BondRx and Discovery Ultra, one can detect multiple splice variants and short targets in a single run. Here we are presenting the workflow for the BaseScope LS assay. Slides are baked in beeswax. Then on the instrument, pre-treatments are applied, followed by BaseScope probe incubation. On the BondRx, the BaseScope workflow continues with various amplification complexes, followed by Leica's red refined kit and hematoxin with the option of offline gluing. For 30 slides, the workflow is about 12 to 13 hours using Leica's software version 4.0 or 5.2. Here is data from the BaseScope LS assay examining splice variants on the BondRx. On the left, we are showing human liver cancer tissues with EGFR probes targeting between exon junctions 7 and 8. On the right is human lung cancer tissue with the MET probes targeting exon junctions 15 and 16. Courtney will be speaking further about these particular examples when she covers the BaseScope application. Also using the BaseScope LS assay in colon tissue, here we're using a short IL-6 probe spanning across 4 junctions. On the right, looking at ovarian cancer tissue with TNF-alpha probe spanning across 3 exon junctions. Here we are presenting the workflow for the BaseScope VS assay on the Discovery Ultra. First, slides are dewaxed. Then on the instrument, pretreatments are applied, followed by the BaseScope probe incubation, followed by the workflow continuing with 8 BaseScope amplification complexes. Lastly, Ventana's red amplification kit is used to develop the red chromogen. For 30 slides, the workflow is about 13 hours using the mRNA universal protocol. Here is data from the BaseScope VS assay on the Discovery Ultra examining splice variants. On the left, we are showing the human liver cancer tissue with EGFR targeting between exon junctions 7 and 8. On the right, in human lung cancer tissue with the MET probe targeting exon junctions 15 and 16. Here with the BaseScope VS assay showing similar data that was just shown previously, looking at colon tissue using a short IL-6 probe spanning across 4 junctions, and on the right looking at ovarian cancer tissue with TNF-alpha across 3 junctions. And now I'm going to turn this presentation over to Dr. Courtney Anderson, who will speak about BaseScope applications.
Great. Thank you very much, Renee. Okay, so there are 2 main areas of application for the automated BaseScope assay. There's exon junction and short target sequence. Exon junction probe design can be used for the detection of splice variants, isoforms, circRNA, gene fusions, and confirmation of gene knockouts. These probes are all 1 ZZ in length. Short target sequence probe design can be used for sequences between 50 and 300 nucleotides, highly homologous sequences, CDR3 sequences for T-cell receptor clones, precursor microRNA, microexons, small nucleolar RNA or SNORNA, and gene editing. These probes can be 1 ZZ in length or more. I will now show some specific examples of how the BaseScope assay can be applied for these 2 application areas. First, we will discuss the exon junction probe design for the detection of splice variants which can be applied for oncology and neuroscience research and beyond. Sensitive and specific in situ detection of cell type-specific splice variants cannot be achieved with traditional methods. RT-PCR and RNA-seq results in the loss of in situ cell type-specific resolution due to RNA extraction from the tissue, and traditional ISH fails to unambiguously identify cells expressing their splice variants, and there are limitations in probe length to determine splice variants. However, the amplification chemistry provided by the BaseScope technology now allows for the in situ detection of splice variants. The MET delta 14 variant is the result of exon 14 in the MET gene being skipped during pre-mRNA splicing, which leads to constitutive activation and oncogenic transformation of the MET receptor. The MET delta 14 variant is present in a wide variety of cancer types, particularly lung cancer. Detection of alterations in MET exon 14 could aid in identifying patients likely to benefit from MET inhibitors. Therefore, we examined the detection of this variant using the BaseScope Automated Assay. To specifically detect the MET delta 14 variant, 2 exon junction probes were designed. One that spans the junctions of exons 13 and 15 to detect the MET delta 14 variant transcript, and another that spans the junction of exons 14 and 15 to detect the wild-type MET transcript. And both probes were tested in 2 FFPE prepared cell lines, H596, which expresses the MET delta 14 variant, and A549, which expresses the wild-type MET gene. And here we are showing the results using the BaseScope VS assay performed on the Ventana Discovery Ultra. Both probes showed mutually exclusive expression patterns, with the exon 13-15 probe only detecting signal in the H596 cell line, and the exon 14-15 probe only detecting signal in the A549 cell line. Furthermore, the probe for DAP-B showed no signal, indicating no background. And here in this slide, we are showing the results using the BaseScope LS-Assay performed on the Leica Bond RX. Again, both probes showed mutually exclusive expression patterns, with the exon 13-15 probe only detecting signal in the MET delta 14 positive cells, and the exon 14-15 probe only detecting signal in the MET wild-type cells. And again, DAP-B showed no signal, indicating no background. Overall, this data demonstrates specific detection of the MET variant, MET delta 14, in situ, utilizing the automated BaseScope assay. The splice variant EGFRV3 is a gain-of-function mutation that arises from an in-frame genomic deletion of exons 2 to 7, leading to constitutively active oncogenic signaling and is associated with a poor prognosis in glioblastoma patients. Detection of EGFRV3 in FFPE clinical specimens by IHC and real-time RT-PCR approaches have been developed but are suboptimal due to the lack of specificity and spatial resolution, respectively. Therefore, a robust assay to identify EGFRV3 status within the tumor morphological context is highly desirable. We use the BaseScope assay to identify EGFRV3 status within the tumor morphological context. 1 ZZ pair probes were designed to span the exon junctions in order to detect either wild-type, mutant, or both transcripts. The wild-type EGFR probes span the junctions of either exons 1 and 2 or exons 7 and 8, while the EGFRV3 specific probe spans the junction of exons 1 and 8. A common probe that spans the junctions of exons 8 and 9 was also used to detect total EGFR, or both wild-type and EGFRV3 transcripts. In this slide, we show 2 glioblastoma cases stained with the probes I just described. With both wild-type probes, we see staining in both cases, indicating both express wild-type EGFR. However, with the mutant probe, we only see signal in the case on the left, indicating that this case is positive for the EGFRV3 variant, while the case on the right is not. Taken together, we can identify EGFRV3 status in situ in FFPE glioblastoma samples. ARV7 is one of 17 splice variants of the androgen receptor and has been implicated in drug-resistant metastatic prostate cancer. But tissue-based in situ detection has been limited by lack of specificity. The BaseScope ISH assay was used to detect exon junctions specific to either ARV7, which is the junction of exon 3 and cryptic exon 3, or full-length androgen receptor, detected by the junction of exons 7 and 8 in metastatic prostate cancer tissues. Shown here from our first publication utilizing the BaseScope assay for exon junction detection are representative tumors from a study demonstrating a range of androgen receptor full-length and ARV7 detection. The researchers found that ARV7 status determined by the BaseScope assay was significantly associated with shorter PSA progression-free survival and showed a trend towards an association with progression-free survival, while AR full-length status was not associated with either PSA, PFS, or progression-free survival. The neuregulin ERBB4 signaling pathway is reported to be associated with a risk for schizophrenia. There are 4 isoforms of the neuregulin receptor ERBB4. Because of the different functions imparted by the distinct ERBB4 splice variants, it is critically important to identify the cells that express these distinct isoforms. Urban et al. Used the BaseScope assay to investigate, with single-cell resolution, the expression of 4 transcripts encoding ERBB4 that result from alternative splicing of exons that alter receptor stability and signaling modes. The specificity and sensitivity of 1 ZZ probes targeting ERBB4 exon junctions were validated in the hippocampus from ERBB4 delta 2 knockout mice, which lack the common exon 2. Signal was detected by fast red, which is visible both in fluorescence and bright field microscopy, as well as DAB. Both pan probes targeting exon 2 labeled scattered cells in the wild-type hippocampus, depicted by the red arrowheads, and generated no signal in sections from the ERBB4 delta 2 knockout mice, confirming the specificity of these probes. Now I'd like to show some data demonstrating the use of the BaseScope exon junction assay for the detection of circRNA, which can be applied again in neuroscience, oncology, and beyond. CircRNAs are a recently identified class of non-coding RNAs that result from the joining of the 5' end of one exon with the 3' end of another, a process termed head-to-tail splicing. They display tissue and developmental stage-specific expression patterns and are particularly enriched in the brain. However, they also play a role in cancer and other diseases. For example, the circRNA CDR3 has been shown in cancer to be a microRNA sponge, and in that way can influence gene expression. However, because of their recent discovery, circRNAs have no clear, well-defined function yet. Therefore, accurate detection and localization of circRNAs are key factors to elucidating their functions. To visualize the circular form of DLGAP1 in situ, we designed 3 BaseScope probes, all 1 ZZ in length, to detect the circular, linear, and total RNA forms. 1 probe to detect the exon 7, exon 9 junction, which is specific to the circular RNA form of DLGAP1. 1 probe to detect the exon 9, exon 10 junction, which is specific to the linear RNA form of DLGAP1. And 1 probe to detect the exon 7, exon 8 junction, which is common to both the circular and linear forms of DLGAP1, which we refer to as total RNA. A Nature Neuroscience article published by you and colleagues in 2015 showed that circRNAs change expression during neuronal development and plasticity. Therefore, we focused on RNA expression changes during development by including wild-type mouse brains from postnatal days 1, 10, and 30 in this study. Postnatal day 10 is a crucial time point included because of synapse formation at this developmental stage. Shown here are images focusing on part of the CA3 region of the hippocampus. We observed signal at all 3 developmental stages for all 3 forms of DLGAP1. While there were some differences in total RNA and linear RNA over development, the most dramatic changes were observed in circular RNA. From postnatal day 1 to postnatal day 10, there was only a slight change in circular RNA detection. But from postnatal day 10 to postnatal day 30, there was a pronounced increase in circRNA detection over this developmental time period. Taken together, this data demonstrates detection of both circular and linear RNA forms of DLGAP1 in the CA3 region of the mouse hippocampus, and correlates with published reports showing changes in circRNA expression over mouse brain development. Here is an example of using the BaseScope exon junction assay to confirm deletion of a circRNA. Kawika and colleagues deleted the circRNA CDR3AF from the mouse genome using CRISPR-Cas9, and then verified deletion of the locus in the mouse brain. The last application area of the automated BaseScope assay I would like to highlight is the detection of short sequences. In this section, we will highlight the detection of CDR3, precursor microRNA, and highly homologous sequences. But there are more application areas beyond this that I mentioned earlier in my talk. The CDR3, or complementary determining region 3, is a highly variable domain in T-cell receptors. Typically, the CDR3 sequences are quite short. For example, the CDR3 alpha and beta sequences from the Jurkat T-cell line are 51 and 48 nucleotides in length, respectively. To identify the specific CDR sequences expressed in Jurkat cells, antisense probes for CDR3 alpha and beta that are expressed in the Jurkat T-cell line were generated, in addition to sense probes for CDR3 alpha and beta to serve as negative control probes. All the probes were then tested in FFPE-prepared Jurkat cells with the assay. Robust staining was observed with antisense probes for both CDR3 alpha and beta in the Jurkat cells, whereas sense probes detected little to no signal. These results demonstrate the ability of the short target assay to discern between highly variable but short CDR sequences for T-cell receptor clones. Retinitis pigmentosa is a form of inherited retinal degeneration that causes blindness in humans. During retinal degeneration, second-order retinal neurons gradually remodel, and this remodeling starts with bipolar cells. Using the RCS rat model to study retinal degeneration and remodeling, the authors of this paper, our first BaseScope paper, which came out last year, performed a microRNA microarray and discovered that miR-125b5p was associated with retinal degeneration, and that it regulated dendritic growth and function in rod bipolar cells. This finding suggested that therapies aimed at reducing miR-125b5p expression could be beneficial in human retinal degenerative disease. To determine the specific cellular expression pattern of miR-125b5p in the retina, the authors used BaseScope probes for the precursor to this microRNA, called miR-125b, and found that the miR-125b signal was present in the outer plexiform layer, internuclear layer, and ganglion cell layer. Furthermore, by combining BaseScope-ish with IHC, they demonstrated that miR-125b co-localized with protein kinase C alpha, a marker of rod bipolar cells, suggesting that the rod bipolar cells could be miR-125b5p positive. Here's an example of a highly homologous sequence. Hutchinson-Gilford progeria syndrome, or HGPS, is characterized by premature aging and loss of subcutaneous white adipose tissue, or SWAP. Mutations in the lamin A gene result in accumulation of progerin in these patients. Revachon et al. Used a transgenic mouse model expressing an HGPS mutation that leads to long-term human progerin expression in SWAP. They called this mouse model rare progerin-expressing, or RPE mice. They revealed that sustained progerin expression in a small percentage of SWAP pre-adipocytes and adipocytes contributes to aging-associated diseases and results in SWAP depletion. The BaseScope-ish assay, used for species-specific detection of human progerin in mouse SWAT tissue, detected low frequency of progerin transcripts in a low number of cells, absence of progerin in wild-type SWAT, and overexpression of progerin in the positive control expression skin. Mutations in C9orf72 are the most common cause of familial ALS. Expansion of a GG-GG-CC repeat in this gene accounts for 10% of sporadic ALS cases. Increase in expression of GLU-A1 AMPA receptor, or AMPAR subunit, occurs in motor neurons with C9orf72 mutations. The precise mechanisms of motor neuron death in ALS is unknown, but excitotoxicity mediated by glutamate and AMPAR GLU-A1 subunits has been implicated. Selvaraj et al. Show that upregulation of the GLU-A1 AMPAR subunit in human motor neurons with the C9orf72 repeat expansion could be the pathogenic mechanism in ALS patients. The BaseScope-ish assay was used for analysis of 4 AMPAR subunits, GLU-A1 through 4, and postmortem studies of control and ALS patients carrying the C9orf72 repeat expansion mutation. The BaseScope assay revealed elevated expression of only GLU-A1 expression in the spinal cord of ALS patients compared to control, but no difference was observed for any of the subunits in the prefrontal cortex. So in summary, I have highlighted two main areas of application for the automated BaseScope assay, exon junction and short target sequence. And now I'll turn the presentation back over to Renee.
Thanks, Courtney. And lastly, to conclude this webinar, I wanted to make the audience aware of our Pharma Assay Services team, which consists of ACD scientists to offer their expertise in running the RNAscope and BaseScope assays, resulting in high quality and consistent results of your stain slides through prospective studies. The team offers a fast turnaround time with scientific guidance, tissue optimization, and data is delivered within an average of 4 weeks. Finally, please take note of our technical support contact information. Please feel free to reach out to us if you have any questions or if we can be of